So when we're testing a brand new medicine or maybe a new surgical technique, how do we really know if it works? How do we make sure that we, the patients, the doctors, even the people analyzing the data, aren't just seeing what we hope to see? Well, that's where the really clever architecture of research comes into play. We're about to dive into the world of blinding and clinical trial designs, basically the blueprints researchers use to uncover the truth.
Here's our game plan. We'll start with the main villain of our story, the bias problem. Then we'll look at the layers of secrecy that trials use. After that, we'll check out the different blueprints or architectures of a trial, tackle a tricky real-world challenge, and then we'll wrap it all up with the ethical bottom line.
All right, first things first. The whole reason we have all these complicated designs is to fight one single enemy, human bias. And trust me, it's a powerful force that can mess up even the best research if we're not careful.
Now you might be thinking, hold on, we already talked about randomization, right? Just flip a coin for who gets what treatment. Isn't that enough to make it fair? And that's a great point. Randomization is awesome for preventing bias at the very start of a trial. It makes sure our groups are balanced. But what about all the little biases that can sneak in during the trial? Yeah, that's a whole other can of worms.
So this is our secret weapon against that kind of bias. It's called blinding, or sometimes you'll hear it called masking. The idea is as simple as it is powerful. If nobody, not the patient, not the doctor, knows who's getting the real treatment and who's getting the placebo, then their personal beliefs and expectations can't unconsciously color the results. It's really the only way to keep the experiment objective.
Blinding is specifically designed to fight two sneaky enemies. First up is performance bias. This is all about how people act. For instance, if a doctor knows you're on the exciting new drug, they might subconsciously give you a bit more encouragement. That extra attention could actually be what makes you feel better, not the drug itself. The second enemy is ascertainment bias, or observer bias. This is all about how we see things. If a researcher analyzing a scan knows you're in the new drug group, they might be just a little more likely to call a fuzzy, borderline result a clear improvement. Blinding just gets rid of all that.
Okay, so if blinding is our solution, how does it actually get put into practice? Well, it's not a one-size-fits-all kind of thing. It actually comes in different levels of rigor, kind of like layers of security protecting the truth.
Think of it like this. Level 1 is single blind. This is basic security. Only the patient is kept in the dark. That's good, but the doctor still knows what's up, which could lead to that performance bias we just talked about. So for the gold standard, you need double blind. Now, neither the patient nor the doctor knows who's getting what. This is absolutely key for most drug trials. And then, for maximum security, you've got triple blind. This is where the patient, the doctor, and even the person analyzing the data have no idea who is in which group until the very end. Each layer just adds more and more protection against bias.
All right, so we've talked about protecting the trial. Now let's shift gears and talk about the fundamental structure of the trial itself. You can think of these as the different architectural blueprints researchers choose from depending on what they're trying to figure out.
The most common, the classic, the go-to design is the parallel arm. And it's pretty much what it sounds like. You set up two or more groups and they just run alongside each other in parallel for the whole study.
The logic here is just crystal clear. You've got Group A on one track getting treatment X. You've got Group B on a parallel track getting the placebo. They run their races, and at the end you see who did better. It's the workhorse of clinical research for a very good reason. It's clean, it's simple, and it works.
But now, let's look at a really clever alternative, the crossover design. With this one, instead of comparing two different groups of people, you have just one group, and every single person gets both treatments. The amazing advantage here is that each person acts as their own perfect control. You're not comparing me to you, you're comparing me on the drug to me on the placebo. This is so powerful that it often means you can get a solid answer with way fewer people in the study.
And here's the elegant little dance it follows. Half the group starts on treatment X, while the other half starts on treatment B, the control. They do that for a while, then everybody takes a mandatory pit stop, and then they switch lanes. The first group now gets the control, and the second group gets the real treatment. Now that little pit stop in the middle, that is absolutely essential. It's called the washout period, and its only job is to make sure that the first drug is completely gone, totally flushed from the person's system before they start the second one. If you didn't have a good washout, the effects would just bleed into each other and the results would be a total mess.
So, what if your questions are more complicated? What if it's not just does drug X work, but also does vitamin Y help? And more importantly, what happens when you combine them? Do they work better together or is it a bad combo? To answer all of those questions at once, researchers use the super-efficient factorial design.
See how this works? By creating these 4 specific groups, you can perfectly tease apart all the different effects. You see what drug X does by itself, what drug Y does by itself, and what happens when you put them together, all compared to a group that got nothing. You're basically getting 3 experiments done for the price of 1. It is a masterclass in research efficiency. So far, we've been talking about these nice, neat, ideal scenarios. But you know, in the real world, things can get messy. Sometimes that gold standard double-blind trial, well, it's just not going to happen.
Let's take this out of the pharmacy and into the operating room. Imagine a hospital wants to test its shiny new multi-million-dollar surgical robot against the standard way of doing things, laparoscopic surgery. Seems like a straightforward question for a trial, right? But there's a big, big problem.
I mean, seriously, how do you do it? You can't. There's no way to blind the surgeon. They're either controlling a giant robot or they're holding laparoscopic tools. They know which one it is. So double blinding is completely off the table. Now what?
Well, this is where researchers get really clever. First off, you can still blind the patient. They don't need to know which type of surgery they had, so their own reporting of pain and recovery isn't biased. That's a good start. But here's the critical step. You bring in blinded outcome assessors. These are independent doctors who weren't in the operating room. Their job is to evaluate the patient's recovery, check the incision, assess their pain, without knowing which procedure was used. This brilliant step neutralizes that observer bias and saves the whole study.
And this brings us to our final and maybe most important section. You see, designing a trial isn't just about being scientifically clever. It's also a huge ethical responsibility. The well-being of the people in the trial has to come first, always. As we saw with the surgery example, sometimes blinding is just practically impossible. The same goes for things like comparing 2 types of talk therapy. But there are also really deep ethical barriers. Let's say there's already a safe and effective treatment for a disease, what we call the standard of care. Is it really ethical to ask someone to join a trial where they might get a placebo, a sugar pill, instead of that proven treatment? In almost every case, the answer is a hard no. You can't ethically withhold a treatment that you know works.
So, the solution is something called an active control trial. Instead of comparing the new drug to a placebo, you compare it directly against the current best treatment. The whole question of the research shifts. It's no longer, is this new drug better than nothing? Instead, it's, is this new drug just as good as, or maybe even better than, the best thing we already have? This is how we make sure every single person in the trial is getting good care while still pushing science forward.
To solidify your understanding, practice the MCQs available on the BCBR app.